In this session, I want to talk about how protein receptors can act to pick up stimuli from the environment and convert that into ion movements that then in turn can cause action potentials. I'll start with mechano-receptors. Here's the membrane and mechano-receptor, a stretch channel that's closed. That stretch channel will be tethered to the membrane either on the inside to actin, the cell cytoskeleton, or on the outside to extracellular matrix proteins. When there's no pressure, that receptor protein just stays closed. But if there's pressure, if something pushes on that membrane, then the receptor gets pulled open. And the result is that the ions that fit through that receptor type can come into the cell in a mechanoreceptor. And those ions, as they come in, they repulse nearby positive charge, and that positive charge can in turn result in somewhere distant opening, for example, voltage-gated sodium receptors. So in this case, what you're seeing is the positive charge near the base of that receptor. That's going to repel this hinged part of the voltage-gated sodium channel, and that will allow more sodium into the cell. And that can start an action potential. What about other receptor types? Another important receptor type that we'll see over and over again are chemoreceptors. Chemoreceptors bind to some molecule. So let's imagine we have some molecule that has a particular shape and charge distribution. It can bind to a chemoreceptor. Here's our chemoreceptor protein in the membrane again. And when it binds, that causes the cell to change shape, the protein to change shape in some way. That change in shape activates, in the case of G protein coupled receptors, G protein coupled receptors, which get their names because the receptor protein is associated with this G protein, and when binding happens, a part of that G protein separates out and interacts with some other protein, often adenylate cyclase that we've talked about before. And adenylate cyclase that's activated can convert ATP to cyclic AMP, adenosine monophosphate. Adenosine monophosphate then diffuses away from that area as it's made, and that in turn elsewhere on the membrane can bind to a channel and cause that channel to open. So that with cyclic AMP bound, sodium can enter the cell. And again, that sodium can start an action potential. You have maybe 1,000 genes for G protein coupled receptors that are involved in chemoreception. Some of them are not active in you because most of them are involved in your sense of smell, and some are pseudogenes. But in your sense of smell, in your nasal epithelia, you express about 500 of these, each one binding to a different shape. And each one, therefore, specific to a particular chemical that fits inside. So that's chemicals, and chemoreceptors are involved in your sense of taste and smell, but you also use them as part of your sense of pain. So nociceptors these are receptors that send action potentials to your pain centers, and they often respond to materials released from damaged cells. So here's a damaged cell that's leaking its contents, and if those contents can bind to a G protein coupled receptor in a neuron that sends pain signals, then that G protein coupled receptor again can activate a second messenger, such as cyclic AMP, that can bind to something that allows sodium into the cell. Many animals detect electrical fields and that's particularly important for aquatic animals living in salt water. For animals like ourselves that are living in air, which is an extremely good insulator, electrical field detection is not especially important. But for aquatic animals, they're using the same voltage-gated sodium channels that we've talked about before, and in an electric field, if we get negative charge outside the cell, that can attract this gate. And once that gate opens up, again, sodium can enter, repel nearby positive charges, and again, start an action potential. And so particular cells in different parts of an animal's body can detect the electrical fields near them and send action potentials to the brain. What about magnetic senses? So a lot of animals have a magnetic sense. Humans may or may not have one. There's some argument about that. And for magnetic sense, what we need is one of two mechanisms. I'll talk mostly about one. Animals, as you know, can make crystals, and crystals of magnetite orient according to magnetic north and magnetic south. So they'll twist until they push in that direction. Well, if we put a magnetite crystal in between a pair of cells and we put voltage stretch sodium channels, so stretch the mechano-receptors that we just talked about, then if the animal turns, if, say, north is this way, then the magnetite crystal will turn, push against this cell, and when it does, it'll pull open those stretch receptors and allow sodium to enter the cell. That means that if this cell is releasing neurotransmitter or is a neuron, then action potentials induced by this cell mean that magnetic north is over here. So stretch receptors are involved in magnetodetection and detection of magnetic north. And there's some evidence for this. In trout, for example, tiny particles of magnetite have been found in an area of the brain where we think these receptors are, and they're using that to detect magnetic north, and it helps them migrate or move around in stream systems. What about hearing? So your sense of hearing involves a type of cell that's called a hair cell. And hair cells have stretch channels in these hair-like structures. They're not true hairs. And if those are bent, if we bend this, then we open these stretch channels, allow sodium into the cell. In the case of hair cells, that causes neurotransmitter release. The sodium, that positive charge, causes voltage-gated calcium channels to open. The calcium enters the cell. The calcium binds to a calcium binding protein, which in turn connects to a vesicle of neurotransmitter, and that causes the neurotransmitter vesicle to fuse with the membrane. The result is neurotransmitter release and receptors in a neuron are activated, and again, either they open ion channels directly if they're ionotropic receptors, or they activate a second messenger. We end up with sodium inside the cell, and again, that can start an action potential leading back to the body of the neuron and the axon.